Electrophoresis is the process of separating large macromolecules, typically DNA, RNA, and proteins. It was originally developed in the 1940s, employing a roomful of equipment, and popularized in the 1950s by Oliver Smithies. Today, it has been refined to a small desktop unit, but it is still somewhat expensive. While high school students throughout North Carolina are expected to learn this process, very few schools can afford a gel electrophoresis kit. DIY biology is a growing movement in which individuals study biology and life science using the same methods as traditional research institutions. These citizen scientists get involved as a hobby or as a nonprofit endeavor for community learning and open science innovation. Recently, the National Institute of Environmental Health Sciences hosted workshops and seminars for science teachers in North Carolina. As part of the workshop, Triangle DIY Biology presented a working session showing a method of creating an electrophoresis experiment using equipment and resources that are readily available and affordable. In this video, we show the details of that working session, demonstrating how to build and run a basic gel electrophoresis experiment using items that you can find around the house or purchase from any grocery store. Agar for the gel can be found in most Asian food markets. For our kit, we'll be conducting food color electrophoresis to look at the separation of food color dyes. Colored food dyes are made of small chemicals of different sizes and different charges, and like proteins in DNA, they migrate through the agar gel when placed in an electrical current at different rates. The first step is to make the gel in which our dye will migrate. In a flask, we mix 100 milliliters of water and 0.2 grams of baking soda. Just swirl the mixture by hand. Then we add 1 gram of agar and swirl again until it is mixed. Place in a microwave oven and heat for about a minute, checking carefully that it does not boil over. Keep checking and return to the microwave until the solution is clear and not granular in any way. Let cool for 15 to 20 minutes. After cooling, we're ready to make the mold. Our mold is made from the lid of a pipette tip box. The gel comb is made using a laser cutter or a 3D printer. Pre-made combs are available online from many DIY biology groups, including Triangle DIY Biology. Place the comb in the mold and slowly pour all the mixture into the mold. Let this set for 1 to 2 hours. While we're waiting for the mold to set, we can prepare the dyes. For this, we use water, corn syrup, and food dyes in a culture plate. Put some water into a beaker or any other container and place 3 milliliters of water in 6 of the reservoirs. Next, add 0.5 milliliters of corn syrup for each. This adds density to the mixture. Lastly, add 2 drops of food dye. You can experiment with different combinations of color, but here we're using 4 single color and 2 mixed colors. For some students, it is also fun to use candy such as Skittles or M&Ms instead of dye. Kids enjoy seeing what their candy is really made of. our mold should be completely cool and firm to the touch. Very gently remove the comb. This creates 6 wells where we will load our sample mixture we just made while we were waiting for the mold to cool. Using a sharp knife or blade, cut the ends of the mold to make room for the electrodes from your power supply. Use the spatula to remove the excess gel and discard. Keep in mind that there is nothing toxic about any of these substances. Now fill the mold with a buffer solution of 0.2 grams of baking soda to 100 milliliters of water, or 0.2%. Make sure the gel and the wells are entirely submerged. Crimp 1 mm stainless steel wires so they fit into each channel we just created in the gel, with the ends facing up. Paper clips are acceptable in a pinch, but they corrode very quickly and stain the mold, so stainless steel is preferred. The power source is a series of 5 standard 9-volt batteries daisy-chained together. Keep in mind the voltage here is extremely low and there is minimal risk of shock. Connect the positive end of the batteries to the wire farthest from the wells with the alligator clips. Connect the negative end of the batteries to the wire closest to the wells. The dye will migrate from the negative charge to the positive charge. Do not connect the negative end to the battery yet. This will be done after we load the dyes into the wells. Draw a few drops of your dye mixture into a pipette and load it into a well. Place the pipette into the well before releasing the dye. It's okay to overload a little since this will diffuse away during the run and won't affect the result. Load each of the 6 wells with one of the 6 dye mixtures that was made. As soon as the samples are loaded, complete the circuit to begin the run. Once the process begins and the power is flowing through the mold, you will see bubbles coming off the wire. That's how you know it's working. Let it run for 1.5 hours. The dyes have a negative charge, so they migrate to the positive end over time. The molecules of the different colored dyes are different molecular weights. The smallest molecules moving faster through the dye than the larger ones. You can see how this separates the various colors from each well. And that's how you can make an inexpensive agar gel electrophoresis kit using everyday items you can easily locate. We hope you've enjoyed this video and good luck with your electrophoresis.